Hello, everyone. What I will talk to you about is speciation. This is the process where one population or species somehow, over time, becomes two. And not just two populations, but two populations that are reproductively isolated from one another. And so this obviously happened in the case of humans and chimpanzees, but this is happening all the time in nature and is responsible for all the biodiversity that we see today. We've thought about this for a long time, more than a century, going all the way back to Darwin, and yet we still don't really have a great understanding of exactly how this process happens. There are lots of models of speciation, some more controversial than others, but quite a popular model is this BDMI model, where we have some population, in this case some frogs, and they become separated in space for some reason. Perhaps there's a mountain range or a river. So now these frogs are geographically isolated and they evolve slightly separately. And mutations accumulate in either population. And so when perhaps this river is drained and they have an opportunity to come together again and hybridize, we'll have combinations of mutations that have never been seen before by natural selection. And it may be the case that these are incompatible, and as a result, you might have very low fitness of hybrids, and they might be infertile or just inviable. And so what this means is that actually we have two reproductively isolated populations. And we have some evidence for this model from nature. An interesting twist on this model is that it's not single mutations that cause incompatibilities, but actually it's chromosome rearrangements. So in this example here, I've shown some chromosomes that have fused together. They've joined at the ends. And over time, this might spread in the populations. Now, all of these frogs have these fused chromosomes, and these have separate ones. And now the hybrids that they might try and form have uneven chromosomes. This can cause problems at meiosis, and therefore aneuploidy in subsequent generations, and therefore poor fitness, and therefore reproductively isolated populations that tend not to have an exchange of genetic material between them. And although I think this idea is quite attractive and lots of people have advocated for the last century, there's actually very little evidence for it from nature. And so what I decided to do in my PhD, or decided to focus on, is whether there is any evidence in this particular system of butterflies. And so these are, really nice species, and I think very beautiful, but what's really interesting about them is their chromosomes. So what I'm showing here is a little tree on the left. Each corresponds to, or each leaf corresponds to one of these four species. And then in the rows, these are chromosomes from genome assemblies. So these horizontal bars represent chromosomes in each species, so in Fabriciana, and then these three Birnstiel species. And the lines that connect these horizontal bars are the same genes in either genome. And so, although this plot might be slightly overwhelming, the take-home message is that rearrangements happen all the time in these species, where chromosomes either fuse together or split apart. And so the question is, have these rearrangements promoted the formation of new species, or are they just coincidental with the speciation process? And so I focused in particular on this pair of species, Birnstiel and Birnstiel daphne, which are sister species to one another. In other words, they are each other's closest extant relatives. And we can compare their genomes like this, where here are genome assemblies I put together where, again, these horizontal lines represent chromosomes, and these vertical lines, or in some case curved lines, are connecting the same nucleotides in either genome. And nature has done, in some way, a very nice experiment for us where half of the genome is not rearranged. They are one-to-one copies, essentially. But then the other half have undergone complex rearrangements where chromosomes either split into pieces or they fuse together, or the same chromosome has fused to different chromosomes in either population. And so these butterflies have gone through some speciation process in time, probably over many, many, many generations, perhaps millions. And then during this time, chromosome rearrangements have arisen and maybe fixed in the population. And perhaps there's also been some hybridization during this process, so gene flow between the two populations, an exchange of genetic material. And we want to know about this process if we're going to test whether chromosome rearrangements have an effect. So how can we do this? We can focus on current day individuals. And if we sample a segment of DNA, so because butterflies, like humans, are diploid, we would sample 2 segments from this individual and 2 from here. And if we trace back the history of ancestry of that small segment of DNA, we can form this genealogy, where lineages merge at common ancestry events upwards through time. So this is the past and this is the present. And actually these genealogies, they tell us about the past history of evolution in these species. In particular, common ancestry events are informative about the affected population size, as in larger populations, common ancestry events are rarer. Additionally, if we trace segments that are sampled from different populations, the time at which they form common ancestry tells us about when the two species diverge from one another. Additionally, if we have events like this, where there's a movement of lineages between populations, this tells us about gene flow, in other words, hybridization, where genetic material is moved from one population to the other. And we can fit a model where our model tells us about population size, divergence time, gene flow, given the genealogies that we observe. The big problem, the really bad news, is that genealogies are unobservable. They do exist, they're a real thing, but we can't see them. We can't even really reconstruct them. So we have to take a different approach, and we can focus on, again, a small segment of DNA, but focus on the sites in this segment where there are mutations, there are differences among individuals. And different types of mutations will correspond to different branches on a genealogy, and the more frequent mutations are, the longer those branches in the genealogy will be. And so now we can do a different type of calculation where we estimate our model now from mutation patterns, or we integrate over the uncertainty in the genealogy that we can't observe. And we don't just do this for one small segment of DNA, but for many, many of them across the genome. In fact, hundreds of millions of these. And from this, we can reconstruct a history for these two species, Birnstiel and Birnstiel daphne, where a large population has split into two about 2 million generations ago. And they split into two quite different sized populations, importantly with gene flow from this one population into the other. So there's been hybridization, but genetic material has only moved in one direction, it seems. We can summarize the rate of this gene flow with this parameter ME, which is the effective rate of migration. This can be thought about as literally migration, so individuals moving between populations, but also natural selection acting against any DNA from a different population that's deleterious. And so we've got some information now about the history of these two species, but we've learned nothing really about how chromosome rearrangements have affected this process. But what we can do is we can fit this same model, but to each chromosome. And this will tell us about how often through time, since the two species diverge from one another all the way to the past, how often different chromosomes move across the species boundary. We can start by doing this with these non-rearranged chromosomes in grey, and we see that there is some variation with chromosome 11, this one here, being an exception. So is there anything special about this chromosome? Yes, this is the Z sex chromosome. So humans have XY chromosomes, but butterflies have ZW, where males have two Zs, and a female has a Z and a W. And there are many reasons that we'd expect a Z chromosome that's hemizygous in females to have selection acting against its transmission between species. What about if we do this same approach, but for the rearranged chromosomes? We find that their rate of gene flow, this ME parameter, is much lower. And so this is telling us that when these two species have hybridized, the bits of the genome that make it across the species boundaries tend to not be these rearrangements. And so it looks like rearrangements are not favored by natural selection and are actually keeping these species apart. We can get an even more detailed look if we apply this same model across a chromosome. So this is Birnstiel chromosome 8, and it's derived from a fusion, a joining of two ancestral chromosomes just here. So this is an ancestral chromosome in yellow, and this in purple. And if we fit the model, we get this map of where genes have been exchanged between the two species over time, and we find essentially a desert around the fusion point. And we can imagine that this chromosome crosses the species boundary, and it will recombine, it will go into smaller segments. The segments that stay in the population tend to be out here and out here, but not around the fusion point, because this rearrangement will be deleterious in that other population, as it will cause problems during meiosis. And we don't just find this pattern on chromosome 8, but also other chromosomes with similar rearrangements. So, in conclusion, this work we've shown that these fission and fusion chromosome rearrangements can promote speciation. It's probably not the only process that's led to speciation in these butterflies, but we can show and quantify that it does have an effect, that actually this is one of the forces that has kept these species apart and is promoting its divergence. But additionally, we've also shown that just from a sample of current day individuals, we can use population genomic inference to reconstruct the speciation process. I'd like to thank my supervisors in Edinburgh, Conrad Loser, Simon Martin, colleagues Derek, Dominic and Roger, these funding agencies, the Max Birnstiel Foundation for inviting me to give this talk. I'd like to thank you all for listening.